to today's public lecture series from Van Andel Institute, Parkinson's Disease from Genes to the Clinic. I'm Steve Ozinga, Philanthropy Director here at Van Andel Institute, and I'm glad you joined us today, but I must admit, I'm ready for the day when we can host these lectures again in person, so hopefully that is soon. Today, VAI scientists Dr. Darren Moore and Dr. Michael Henderson are joining us to provide a deeper look at their area of expertise, Parkinson's disease. This complicated disease has an equally complex group of factors that contribute to its onset. Age, genetic risk, and even our individual brain structures play a role in the origins of Parkinson's, but these also offer hope for new therapies that could slow or stop disease progression. At the Institute, Doctors Moore and Henderson explore factors that can contribute to a person's Parkinson's risk and are also in search of breakthroughs that could one day be translated into life-changing treatments. If you have questions about anything during today's presentation, please drop them in the chat function or hold them till the end when we do a Q&A session. So let me introduce our expert speakers today. First of all, Dr. Darren Moore is the department chair and a professor in VAI's Department of Neurodegenerative Science. He seeks new treatment approaches for Parkinson's by investigating the inherited form of the disease, which makes up 5 to 10% of cases. He also aims to translate the understanding of genetic mutations into better treatments and new diagnostic tools for Parkinson's. Dr. Moore received his undergraduate degree from the University of East Anglia and a PhD in molecular neuroscience from the University of Cambridge. He joined Johns Hopkins faculty in 2005, became an assistant professor in 2006. He moved to the Swiss Federal Institute of Technology in 2008 and made his way to Grand Rapids and Van Andel Institute in 2014 when he joined the faculty as an assistant professor and was promoted to professor in 2017. Up next is Dr. Michael Henderson, an assistant professor in VAI's Department of Neurodegenerative Science. Dr. Henderson investigates the cause of diseases like Parkinson's and dementia with Lewy bodies, along with the factors that control disease progression. He hopes to translate his findings into therapies that could slow or stop progression. Dr. Henderson earned his bachelor's in biological science from Florida State University, followed by his PhD in neuroscience from Yale University, and following his doctoral research and postdoctoral fellowship, Dr. Henderson joined VAI in 2020. So thank you again for joining us today and for your attention, and let's get the program started, and I will turn it over to Dr. Henderson.
Thanks, Steve, and thank you all for coming today. I want to welcome you all to our public lecture series at Van Andel Institute. My name is Mike Henderson, and I'm an assistant professor at Van Andel Institute in Grand Rapids. And today I'll be discussing Parkinson's disease from genes to the clinic, and I'll tell you about research being done at the Institute, including my own lab, and our focus on Parkinson's disease pathology. My group investigates the underlying pathogenesis of neurodegenerative diseases and leverages that knowledge to develop and evaluate therapeutics for these devastating diseases. One of the ways we do this is by closely and quantitatively examining pathologies associated with disease, and this is one of the things that I'll talk about today. Neurodegenerative diseases are progressive disorders that affect the nervous system of patients, experienced as motor or cognitive dysfunction that's debilitating to individuals with disease. Two of the most common neurodegenerative diseases are Alzheimer's disease and Parkinson's disease. Alzheimer's disease is a form of dementia that interferes with memory, thinking, and behavior, and these symptoms are generally progressive, leading to disruptions in daily life over time. Parkinson's disease is a motor disorder, and patients may experience symptoms including tremor, difficulty balancing, and difficulty walking. Parkinson's disease patients can also exhibit seemingly unrelated disturbances in gastrointestinal function, loss of sense of smell, and eventually dementia. We don't have a complete understanding of what precipitates these diseases. Years of research have begun to uncover their underpinnings. We know that each disease is associated with the misfolding of normal brain proteins into pathological forms that are toxic to neurons. We have these proteins in our brain, all of us do, And it's a bit like having reams of paper in your house. They can be very useful for writing on, printing, making signs, but when they're misfolded, it's like the paper's crumpled up. It's no longer useful, and our brains try to discard it as waste. But over time, our brains are less good at discarding this waste, and they accumulate in elongated fibrils that can eventually kill neurons. You may have heard of some of these misfolded proteins before. In Alzheimer's disease, the brain accumulates amyloid beta plaques and tau tangles. In Parkinson's disease, the brain accumulates alpha-synuclein Lewy bodies. If we take a look through a microscope at a Parkinson's disease brain, which is one of the things we do in my lab, we see many beautiful and presumably healthy neurons in green scattered around, critical to the brain's function. But we also find a subset of these neurons that contain large accumulations of those long fibrils of misfolded protein, known as Lewy bodies. You can see these in magenta. When they are located in the axons of neurons, we call them Lewy neurites. And if I take away the image of the neurons, you can see that this pathology is quite pervasive, filling up the brain and disrupting brain function. We also know that the location of this pathology is important. The accumulation of these proteins is associated with dysfunction of neurons in that region of the brain. So if those neurons control memory, there's memory dysfunction like we see in Alzheimer's disease. If those neurons control movement, there's motor dysfunction like we see in Parkinson's disease. Further, the progressive symptoms of these diseases are related to the spread of protein pathologies through the brain, affecting more neurons and disrupting additional functions. So, how does this process begin? We're still working to find the answer to that question. What we know is that some of the risk of developing disease is carried in your DNA, known as genetic risk. There we go. Other risk is related to lifestyle choices and environmental toxins encountered by individuals during life. Researchers at Van Andel Institute are working every day to understand and develop interventions for treating Parkinson's disease. Faculty within the Department of Neurodegenerative Science at Van Andel Institute are addressing several critical questions in Parkinson's disease related to genetics, environmental toxins, epigenetics, cell biology, neuron circuits, animal modeling, and facilitating clinical trials. I told you that some of the risk, we think less than 25%, is carried in our DNA. And Darren, Jose, Jerry, and Rita are trying to find out which rare and common differences in our genes affect lifetime chance of developing disease, and how. Lena, Patrick, and I look at several certain environmental exposures such as bacteria, viruses, and chemicals that may increase risk of developing Parkinson's disease. And we try to determine how these different molecules affect the brain. One way the environment can affect us is by changing which genes in the body are expressed and which aren't. This modification of DNA is called epigenetics, and it's what makes our heart, and our brain, despite the fact that those two different organs have the same DNA. Jose, Rita, and Jerry look at how the epigenome is changed in patients with disease. Cells are the fundamental building blocks of life. By drilling down to examine processes that occur in cells, we can estimate disease processes at a fundamental level. We can also screen thousands of potential therapies in weeks with this type of system. Cells do not exist by themselves. This is especially true in the nervous system, where the integrated activity of thousands of neurons is necessary for even the simplest movement or thought. Groups like those of Hong-Yuan and myself are investigating the vast network that is the brain and trying to understand things like, how does pathology spread through the brain? And can we restore normal function by modulating those circuits? To ask more complex questions such as these, we need more complex models. So in some cases, we use animal models to understand, for example, the intersection of genetic risk and environmental risk, and to evaluate therapeutics. And when we do find something that looks promising for potential therapeutic development in Parkinson's disease, we can facilitate its path through the clinic through clinical trials. While VAI does not run clinical trials, it does facilitate them through the Linked Clinical Trials Initiative. This initiative takes our best ideas for targets from our research and finds drugs that have already been approved for other conditions and proven safe and repurposes them for Parkinson's disease. Together, these strategies give us a broad perspective of Parkinson's disease and a great ability to push new, exciting research forward. Some of that research is happening in my lab, so what are we investigating? 4 main questions we're researching now are: how do protein pathologies spread through the brain? How do genetic risk factors impact disease? What cellular pathways are disrupted by protein aggregates? And how can we modify disease through therapeutic intervention? In recent work, we found that Alzheimer's and Parkinson's pathologies spread through the brain by following neuronal connections between regions. However, some regions along the route are resistant to pathology. We use quantitative pathology and network modeling to understand what makes certain regions more resilient or more vulnerable to developing pathology. We also look at individual neurons under a microscope to determine how mutations in genes like GBA1 and LRRK2 can change the way that neurons respond to pathological proteins. For example, genetic mutations may reduce the ability of neurons to dispose of pathological aggregates, leading to an overflowing trash bin. Or they may change how quickly proteins spread between regions and accelerate disease progression. We can also examine the contents of the trash bin of cells to find other proteins that are mistakenly discarded. We may be able to use this knowledge to develop therapies for disease. Once we know what's going wrong in disease, we can then target therapies to these pathways. We can reinforce exhausted cellular pathways, reverse damage caused by genetic risk factors, slow or stop the spread of pathological proteins through the brain. Our hope is that by understanding the underlying biology of disease, we can target therapeutics, we can support our body's defense mechanisms, and we can slow or stop the progression of these devastating disease. In conclusion, the brain is a wonderful network of billions of neurons. Those neurons must act together and communicate to form a thought or initiate a movement. When proteins misfold and cellular function is perturbed, whole circuits are disrupted, resulting in symptoms we see as neurodegenerative disease. Scientists at VAI are working every day to discover how this process starts and progresses with the goal of stopping it. I appreciate you attending. I'll be happy to answer your questions after the lecture, but now I'm excited to turn the floor over to Dr. Darren Moore, the chair of our department and a fellow neurodegeneration researcher, to tell you how we conduct studies in the lab and to describe an exciting new program we're developing to leverage our knowledge and our Western Michigan community in the search for answers. Thank you.
that this is more prevalent in males than females, Parkinson's disease. We also think about exposure to things in the environment like toxins and chemicals, so pesticides, traumatic brain injury if you've ever had a concussion or multiple concussions. And as Mike mentioned, some researchers in the Institute are thinking about things like infections with bacteria and viruses as potential triggers of disease. So we can think about the environment contributing many kind of detrimental factors that build up over time, but there are also things that have been identified that are protective. So we know that tobacco usage is somehow strangely protective in Parkinson's disease. Caffeine consumption is also protective at some level. And things like non-steroidal anti-inflammatory drugs like ibuprofen are also showing some sort of protective effect. These things aren't definites, right? So they just contribute in a very small way. So I wouldn't recommend that you do any of these things for protection. Certainly avoid pesticides if you can, and infections are bad in general, right? So, but we don't really know. The answer is we don't really know. We know that combinations of these things epidemiologically are associated with the disease. What we also know is that genetics contributes to disease as well. So your genetic background, we're all unique. We all have the same genes, but those genes vary in very subtle ways in terms of their sequence. So in terms of Parkinson's, we tend to think about it in terms of what we call sporadic disease, where you don't have a family history, we don't really know the causes, or familial disease, which is typically inherited disease, inherited within a family, and normally caused by a mutation in a single gene. So these familial cases are very similar to the sporadic cases. It's really one big disease, and about 10% of those cases are familial. So in some cases, we know the genes involved, and then we can study those genes in the lab to try and understand, how do they contribute not just to the familial disease, but how do they contribute to the sporadic disease as well? That's really the goal here in our institute, is to understand how genes contribute to the entire disease. But we use these windows where we find these mutations in certain genes to understand what they may be doing to individuals. There are also kind of another way of thinking about genetics, right? So you have 10% of cases which are familial, but you also have genes, many genes in this case, what we call multigenic or multiple genes that act as risk factors. So each individual gene has a little variation in it. And alone, this is not sufficient to cause disease. But if you have this variation in many, many genes over time, this can slightly increase your risk of developing disease. Again, it's probably not sufficient alone to give you the disease, but it's one of these major risk factors. The third risk factor for disease is age. This is an age-related disease, and as you age, your chances of developing Parkinson's increase quite dramatically. So we typically think of Parkinson's as an age-related disease that normally in sporadic cases develops between the ages of 55 and 65 years of age. So kind of a disease of older age, but certainly there some familial cases happen at a much younger age, and that tends to be quite typical. So these factors combined, environment, genetics, and age, and possibly other things we're not even aware of at this point, really contribute to your risk of developing diseases and the causes of the disease. So in terms of genetics, kind of, like I said, we can break it down into familial disease, so disease which is inherited in families. And if you have the mutation in a certain gene, you will likely develop disease in your lifetime. And what we call sporadic disease, which is just an individual with no family history. We don't really know why develop disease. And so there are a number of genes now which have been identified, and we can kind of break them down on this interesting chart, which on one axis you can see you have the risk of Parkinson's disease going from low risk to high risk to also causal. So that means it's not risk at all, it's actually causing the disease. And on the other axis of this graph, we have what we call frequency in a population of these mutations, or what we like to more commonly say is these variants. Okay, so very rare mutations to very common variants. We all have variation in our genes, so this is something we all have, but we don't all have genetic mutations which cause disease. These are something that's very, very rare in the population. So you can see up here on the top left, we have genes mapped out in this reddish color, which are very, very rare in the population. And if you have a mutation in one of these genes, this will actually cause PD in your family. So this is the familial disease that we look at. On the other end of the spectrum, you have this kind of greenish box, which is in the lower right-hand corner, which has a very low risk of disease in those via those genes, but they're also very common in the population. So we have a bunch of genes which have these normal common variants in the sequence. They're very low risk factors. So alone, they're not sufficient to give you disease, but they do contribute to the risk of sporadic disease. So in green, we have more of the sporadic factors. In pink, we have more of the familial factors. But then we have things in between as well, like the GBA gene and the what we call the LOCK2 gene. These are genes which, there are variants in these genes are kind of more common, and they increase your risk to something in the kind of medium to high range. And so what you'll notice from this graph, which is very interesting for us at least, is what I was kind of saying at the beginning. Like, we study these 10% of familial cases to really try and understand the greater, sporadic Parkinson's disease, the more common form. And you notice here things like the LRRK2 gene, LLRK2, is in the red group, in the green group, and also in the blue group. So this is a gene which, if you have a mutation, you get familial disease, it's inherited. If you have variation in this gene, it contributes to sporadic disease, and then everything in between as well, right? So you can have other types of variants which just increase your risk more than sporadic disease. So these, we think in the Institute, become great targets for therapies, okay? So for instance, in my lab, we're working on this LRRK2 gene, and many pharmaceutical industry companies are interested in LRRK2 as a therapeutic target for Parkinson's. So there are now drugs out there, what we call inhibitors, that actually turn this gene off. And we've tested them in animal models, and they're actually protective. They protect against neurodegeneration. Now these drugs are actually in phase 1 clinical trials in Parkinson's patients for safety. Okay, so this is the type of thing that that we tend to work on here, try to build the evidence why a mutation in one of these genes causes disease, and what do we really need to do to that gene or the protein that it encodes to actually develop a therapeutic, a targeted therapy. So in the Institute, we work on these 3 genes here at the top, VPS35. Mike already mentioned the alpha-synuclein gene, the protein that aggregates, and then LRRK2 is another gene that we focus on. And these all cause what we call a dominant form of disease, which means if you have one mutation, one chromosome, you would get disease in your lifetime. We also work on LRRK2 gene as well, and we also work on these common variants that we call risk factors for disease. So Jerry Kutzia's lab is kind of the one doing this as well. So Mike introduced you to the faculty already. So we recently rebranded ourselves as the Department of Neurodegenerative Science. And our major focus here now is always on Parkinson's disease. We have a lot of critical mass in this area, but we're also expanding into different forms of dementia, such as Alzheimer's disease, dementia with Lewy bodies, which Mike mentioned as well, frontotemporal dementias. The reason being is that many of these diseases share commonalities. So for instance, dementias overlap very closely with Parkinson's disease. You can have diseases that almost look kind of somewhere in the middle between them. For instance, Parkinson's disease patients commonly develop dementia, which looks a little bit like the dementia you would see in Alzheimer's. But there's also diseases that kind of span the spectrum of dementia and Parkinson's, things like dementia with Lewy bodies. It's a movement disorder, but it also has dementia as a primary symptom as well. So studying all of these diseases together is actually kind of very beneficial to understand common mechanisms. So our department, essentially, Mike already did a great job at covering, what we work on and why. Just to kind of reiterate that, so faculty here in our department are really involved in identifying these genes that are linked to disease, and this is a really core part of what we do. So we're interested in identifying in these inherited mutations which cause familial disease, and this is not just Parkinson's, but this is across neurodegeneration in general. But we're also interested in how these common variants in genes can impact sporadic Parkinson's, so what we call genetic risk. So that's something we do here. This is Jose Brass, Jerry Kutzia, and Rita Guerrero. They work in this area. Then once you know what the gene is and you know what the mutation looks like in that gene, you can then study the mutated gene and the normal gene and try to compare them and figure out, what's the mutation doing differently from the normal gene? What function does it add, or what function does it take away of that gene? So we think about this in terms of disease mechanisms. So how does a mutation cause a disease? And this can impact anything from how genes are regulated to molecular pathways in cells, particularly neurons, such as cell signaling pathways, cascades of signaling molecules, but also larger parts of the cell, which we call organelles, things you've probably heard of like mitochondria and the nucleus and Golgi trafficking, these types of things. These large organelles, these large centers within cells, which have very defined functions. We also think about disease mechanisms in terms of, how are those impacted? And we do all this in different types of model systems. So we use human cells and rodent cells, either cells from the brains of animals, but also we use human cell lines derived from tumors. But we also have this unique tool that's emerged over the last 10 years called human iPSCs, and I'll tell you a little bit more about that towards the end of the talk. But we also rely on animal models, particularly rodent models, because they have a brain just like humans, and so there are some cases where we need to study an actual intact brain and how it affects the behavior of that animal and the ability to move properly. So going from kind of genes to kind of the mechanisms of those genes, we also try to overlay those things on the actual brain itself. And so Mike already mentioned how we tend to look at the effects of some of these genes and proteins in terms of neural circuits. The brain is essentially a large electrical organ that communicates through neurons via electrical currents. And we try to understand exactly how it impacts those functions. And we also think about in our department how we can take some of these genes and the mechanisms that we find and actually start developing or testing therapeutics in some of our systems. So Patrick's lab and my lab and also Mike's lab work on some of these preclinical animal models where we have things like the LRRK2 gene, where we have an inhibitor or a drug, and we test them in these animal models as a kind of a proof of concept to make sure that they would be safe and they would actually work well in humans. Along with therapeutic studies, we also need to be able to track the disease over time in humans. And so some of our faculty work on biomarkers, and these are essentially anything that you can measure in the body that tells you, that you have, say, Parkinson's disease and how the disease progresses over time. So when we think about biomarkers, there are a few that spring to mind, which is things in blood that we can monitor over time that track with the disease, and also imaging of the brain is another way of someone who's still alive but has imaging on their brain to see how the disease is progressing. Lena Brunden's lab here works more on blood-based biomarkers of Parkinson's and how we can use them to track disease. So you can see from our department, we really cover many different aspects of the disease, from all the way from identifying brand new genes through their mechanism to understanding how we can then target those genes for therapeutics. Okay, so next I just want to tell you about some interesting and exciting kind of team science that we're doing, where we all kind of, we all work individually in our labs. We're all the heads of different labs and we lead labs, but we sometimes come together and, scientists love to collaborate and assemble around key questions in the field. So one particular project we have is what we call the IPARC collaboration team. This is Patrick Brunden, Lena Brunden, Mike Henderson, and Andrew Pospisil. And this was recently awarded in this summer, $2 million over 2 years. So this is substantial funding for us. But the idea of this project is to study the role of infection. And I mentioned there are environmental causes of Parkinson's disease. We don't clearly understand how those work. But one idea this team has is to take infections as a particular insult and say, okay, if you're exposed to different infections as a single insult, is this enough to drive the development of Parkinson's disease in your lifetime? And the way this is done is normally with animal models, because they have a short lifespan and we can't really do these things in humans very easily. But here we're taking viruses like hepatitis C, E. coli, and Helicobacter pylori, which is linked to stomach ulcers, and really exposing these animals to these pathogens and see whether they develop disease in their lifetime. But the other way we can think about pathogens is that most of us get sick in our lifetime, doesn't really cause us any major detrimental effects. But maybe pathogens really just act as primers. Okay, so maybe you have an infection and it's fine and it resolves, but it sets up a kind of cascade of events that makes you more sensitive to a subsequent insult that may cause or be linked to Parkinson's. You can imagine in your lifetime, you may have many infections. You may have the flu, you may have COVID. I guess we have to say that today, right? You definitely may have COVID. You may have many things. And so you're going to accumulate infections over time. And maybe it's the accumulation of these things which really, increases your risk of disease. This is just one hypothesis that we're testing in the department. And another recent team-based science initiative is what we call this, a new foundation called the Aligning Science Across Parkinson's Initiative, otherwise known as ASAP, which I think is quite a fitting term for thinking about therapies for Parkinson's. And so this is a brand new foundation developed by Sergey Brin, the founder of Google. And he's basically given up $1 billion to fund a large number of collaborative teams to come together on key questions. And so far, it's 21 teams that have been awarded these grants. And so we have a team involving myself and Jose Braz here at the Institute, in collaboration with our colleagues at the University of Minnesota, Mike Lee and Laura Niedenhofer, where we really come together to answer this question about senescence and the role of cellular senescence in Parkinson's disease. So this was awarded in 2020. Is to address a question about the biology of genes. There were 21 teams funded in the first round. We were awarded 6.2 million of the 3. This is a large team effort to address like a really fundamental problem in Parkinson's disease. And essentially, cellular senescence is a process that happens to all of our cells in the body. And this is really, as you know, all cells have the capacity to divide, or at least most of them do. But cellular senescence is the process by which cells stop dividing, okay? But they're also kind of protected. So once they stop dividing, this senescent process, they're protected, and they basically, they start to secrete factors which are actually detrimental, okay? So cellular senescence is typically considered a detrimental effect. And it's been linked quite closely to aging and age-related processes. So as we age, cells become old, they stop dividing, they start, a normal cell can start kicking out different factors, which makes the cell and the surrounding environment of the cell kind of toxic, okay? So there are many triggers for these senescent cells, like DNA damage being a major one, and they can also regulate a large number of processes, such as inflammatory diseases and the ability of stem cells to continue dividing in the body. So it's really a balancing act in the body between, a normal cell and a senescent cell. This kind of idea of when does a cell become senescent and converts from normal. And like I said, these are linked to many diseases that we know of, particularly Alzheimer's disease, but nobody's really looked at this in Parkinson's. So we hope to be the first team to do that. And the way we're going to do this is to rely on a natural process of DNA repair. So genes get damaged all the time in cells, but they're rapidly repaired by machinery. And if we take away this machinery from, say, an animal model or a mouse model, we can actually induce this idea of spontaneous DNA damage, which will induce senescence in these cells. And therefore, we can see if it's sufficient to cause Parkinson's disease and whether we can remove those cells. So senescent cells activate a large number of processes. Like I said, they stop dividing. This is what we call cell cycle arrest. But they also secrete a large number of factors, which makes the environment around them much more toxic. What we hope to do is address a few different questions, which is in the brain, which types of brain cells? There are many types of cells in the brain, many types of neurons, and also cells we call glial cells, which are astrocytes, microglia, oligodendrocytes, and they all contribute to the health and function of the brain. So we hope to understand, which types of brain cells become senescent in the lifetime of a Parkinson's brain, both a human brain as also an animal model brain, and when does this happen? And then we have some really intricate methods for being able to remove these senescent cells from the brain using pharmacological tools, but also genetically as well. So we're hoping that we can try to understand whether removing these senescent cells during aging will actually protect the brain from Parkinson's. So that could be an interesting therapeutic option in the future. Okay, so the last thing I want to tell you about is a new program which we're developing. I'm sorry about my slides; they're all sort of scrambled up. But we've, through the sort of generosity of the Van Andel family, there's a new endowment fund which has recently been created called the J. and Betty Van Andel Catalytic Fund for Neurodegeneration. And this is a generous gift from the Van Andel family. And basically, we've used this fund to develop what we call the MIND program, so the West Michigan Neurodegenerative Diseases program, which hopes to study the genetic architecture of Parkinson's disease in West Michigan for the first time. Okay, so there really aren't very many studies done on this particular population of West Michigan. We hope to use local clinics to be able to understand exactly what the genetic risk looks like in this region, and also use it for biomarker discovery. There's also a complementary program to this called the MENU program, which is another independent gift you're probably going to be hearing about in the future, which is to address the role of metabolism and metabolic disease in the research around those two diseases. So there's the MIND and the MENU program, which are now these new endowed gifts. So what we hope to do is essentially tap into the local patient population of West Michigan. And what we want to do is create a biorepository of biospecimens from patients and people that don't have disease as well. We're also going to start a new brain bank where we take brain donations from people that are, when they die, will they donate our brain to us? So we're going to build the infrastructure for this, and Mike Henderson is leading this initiative. I'm going to tell you a little bit about this iPSC platform, where we're also developing a new tool to be able to study the mechanisms of disease. And then part of this program as well also supplies grant funding to people within our institute, faculty and postdocs that basically want to apply to work on neurodegenerative disease. And this will be good for spurring research between the different labs. So in terms of the biorepository, what we hope to do is to take mainly body fluids from patients. And when I say body fluids, I mean specimens such as blood, but we're also hoping that we can take urine as well as CSF, which is cerebrospinal fluid that you take from the spine because it's in close contact with the brain. These are kind of the major samples we'd like to collect from patients. So these would be people with not just Parkinson's disease, but dementia as well, different forms of dementia. And really, this is a broad program tackling neurodegeneration in general, but Parkinson's will be a key part of it. So we hope to take blood from up to at least 2,000 people in the first year. And then from this blood, we hope to take DNA, extract DNA, and sequence that DNA. And so we're going to be able to kind of find new mutations that cause disease, but also new risk factors, new genetic risk factors that maybe are linked to disease in these regions. So we can understand a little bit more about what drives the disease locally. And so we're hoping to do this in collaboration with all our local healthcare partners, particularly with Spectrum Health, who we're kicking this initiative off with, and but also everybody locally who sees different patients with Parkinson's disease and dementia. And so the idea is to create what we call a biorepository or a biobank, where we can bank all these samples and use them for research. So people can use blood samples and things derived from blood. They can use CSF, they can use urine and other types of body fluids as well for biomarker discovery. So we can understand, things that are actually, biological effects that are linked to the disease and contract the disease over time. This is particularly important for clinical trials and monitoring the effectiveness of a drug in a clinical trial. But what we really want to do is use it to spur genetic research locally and understand, some of the drivers in this local population. Linked to this, we're starting a new brain bank as well, or a brain biorepository. And we hope to be collecting brains eventually from Parkinson's patients, but also dementia with Lewy bodies and Alzheimer's disease. And then eventually, I think we'll scale up to other rarer diseases as well that are linked to those ones. So Mike Henderson is leading this initiative, and I apologize if this is a bit too small for everybody to see. But what we hope to do is we've partnered with an organization called LifeNet initially, who will provide us with brains from patients that die, post-mortem brains. So we hope to extract the brains from them, have them delivered here, essentially, where we will then divide up the brain into a number of different sections and catalog all those things and barcode them. And then we hope to have them neuropathologically diagnosed so we can understand, if this person had Parkinson's disease, does their brain say that they have Parkinson's, or do they have something a little bit different? So we hope to take that pathological information to give them a diagnosis post-mortem. And then we'll use these brains for studies in our department, basically for extracting DNA and RNA to look at gene expression, for sequencing of those patients, but also, as Mike's research does, for extracting proteins from the brain that have this unique ability to kind of spread pathology from region to region and neuron to neuron. So that's what we hope to do with this brain. So this is a really vital resource for us. It's made possible by this endowed gift from Van Andel. So again, this will be eventually in partnership with local clinics that have movement disorder specialists as well as dementia clinics. And yeah, so I already mentioned, why we need these brains. They're really fundamental for the type of research we do. And finally, one nice way we can study neurodegeneration from patients that are still alive, which is what we hope they would stay alive, but we want to study their disease. So if we can make a biorepository where we collect genetic information from people, and also we have a brain donor program, there's also a really nice, unique model we can use called induced pluripotent stem cell technology, where we can take skin cells from patients, or really any type of cell that you're willing to donate. It could be a blood cell, could be a skin cell, and we can actually turn those cells into stem cells. Okay, so here on the left panel, what we're showing is a patient with disease versus a patient that's disease-free. We can take a skin biopsy, very simple procedure. We can do this in one of the local clinics. And so if we find genetic mutations that are really interesting or risk factors that we want to study further, we can take those cells, we can convert them into stem cells where they can divide indefinitely forever. And so, like back to an embryonic state, like in an embryo. And then we can actually, the unique property of stem cells is we can turn them into any other cell in the brain. So as you can imagine, we can only study the brain post-mortem, right? So this is a bit of a problem. We don't know what happened beforehand during the disease course. So now what we can do is take these skin cells, turn them into these iPS cells, and then eventually turn those into cells that we're interested in, like brain cells, like neurons, glial cells. And then we can study the mechanisms of disease in those cells. So these are cells from patients where we can try to understand exactly what's happening and what's going wrong with a particular genetic mutation. So hopefully what we want to do with those cells is what we call phenotyping, which is to compare, healthy control cells with disease cells, and actually understand a little bit more about, what's going wrong in those cells at a cellular level, a molecular level of gene expression. And then what we hope to do is use those cells as a platform for drug discovery, right? So what we want to do is create this new screening facility where we can go in with these cells and we can actually throw a lot of different drugs at them and basically find drugs or select drugs that protect against some of these detrimental phenotypes, as we call them. And so this is how drug discovery can be done with human-derived cells. And that's what we're trying to create here with our MIND iPSC platform. So we've already recently recruited a world-leading director for this platform. He'll be coming in from Sweden, so he's going to direct this. And we hope to kick off this initiative later this year. So with that, I will just thank my lab briefly for all the research they do, and all the department faculty for everything they do for neurodegeneration. So with that, I think Mike and I are happy to take any questions, and thank you guys for listening. Oh, one final thing I wanted to do before I go is a quick plug. So this lecture was called Parkinson's from Genes to the Clinic. And next week we have the Grand Challenges in Parkinson's Disease annual symposium, which will also be on the same type of theme. So from Genes to the Clinic, specifically focusing on two genes that we work on quite a lot here, this LRRK2 gene and the GBA gene. These are major risk factors for Parkinson's disease. So if you're interested in any way, you'll learn a lot more about the biology, and also there's a great clinical program where we can understand what clinical trials are happening and which ones are being planned. So with that, thanks for listening.
Great. Thank you, Dr. Henderson. Thank you, Dr. Moore, for those fantastic presentations. I had a couple of questions that have come in, Dr. Moore, about the MIND and the menu program, the biorepository. You mentioned later this year, which I know it's October 1 tomorrow, later this year, some of the stuff is going to be up and running, but how long before all of the features and benefits are up and running where people can, drop off samples through their hospitals and those types of things? What kind of time frame are we looking at for that?
Right, yeah, I should have said that. That's a great question. So we're hoping to kick this off in October with Spectrum Health to begin with, and then we're in the process of trying to negotiate agreements with all the other health partners locally. So we think definitely in the next 3 to 6 months, this will just be a mainstream thing for most clinics around here that see most major health partners that have movement disorder clinics and dementia clinics. Yep.
Fantastic. And there's one other related question to that. Are we able to utilize the bio data that's collected through the Fox Foundation's PPMI project as well?
Actually, anybody can access that data for their research. So that's one of the beauties of the Michael J. Fox Foundation. It's widely available. The problem is it's not local and it's not very many cases. So it's a great resource, but we hope to do something very similar to PPMI locally. That's kind of
Yeah, and just to reiterate what you said, a study, I mean, this type of infrastructure is not in place in West Michigan, and this type of thing has never been done in West Michigan before.
No, it's a really unique opportunity, and you should know we've been trying to build this for the longest time, ever since I've been here. This was always part of the plan. This this new gift just made it possible to just go for it. And yeah, it's going to be a wonderful resource both for patients locally to be involved in our research, but also for us as faculty and our labs to really study a local population rather than just a generic kind of national population. So I think that's going to be very useful.
Good. Fantastic. Thank you. And again, to those attending, if you have questions, please feel free to put those in the chat box. Question for Dr. Henderson about timetable for potential therapies. You both mentioned therapies that are in process. What are some timetables of research and actually getting that to a therapy that a patient could use?
Yeah, so I think one of the reasons that we're all doing this research is because there are no disease-modifying therapies for Parkinson's disease or many of these other neurodegenerative diseases. One of the reasons for that is that it takes a really long time for something that we've discovered in the lab to be, tested really thoroughly preclinically and make it through 3 phases of clinical trials. I would say on generally it's probably 10 to 15 years from when we discover something to when it might actually make it to the clinic. One of the things that's really exciting, I just looked it up, there's 142 ongoing clinical trials for Parkinson's disease. About 60% of these are for symptomatic disease, so trying to alleviate those motor symptoms. We already have L-DOPA as one of the major therapies that we use for that, but there's other ways to deliver that and make sure it's more consistent over time. So 60% of therapies in clinical trials are for that. 40% of those are for disease modifying. That is therapies that we think can actually modify the progress of disease in some way. And there's been promising clinical trial results, but as you know, there's no disease modifying treatments yet. I would say we're all hopeful that some of these that are in phase 2 or phase 3 clinical trials might make it into the clinic, but we just have to wait and see. I think some of these will read out in the next few years, so it's a lot of excitement in the field that some of these may prove to be disease modifying.
Great, thank you. Dr. Moore, anything to add to that?
No, I mean, well, maybe. So we're all involved in research, which for most of us has ended up in a clinical trial. So this is quite gratifying, I think, to see that some of the genes, mainly the genes that we work on, are the disease modifying therapies, and they're coming through. I mean, it's 10, 15, 20 years worth of work to get to this point. It's a hard slog, I would say, but it's looking good right now. We have drugs in the clinic that they can make or break Parkinson's at this point. And then let's not forget as well, we also have the Linked Clinical Trials Initiative, which Patrick Runda is involved in. And so this is the repurposing of drugs that are approved for other diseases with the hope that maybe they have some beneficial effect in Parkinson's. This is a kind of a unique, non-targeted approach, but it could be very useful. So, I'd say watch this space, and yeah, we all have our fingers crossed. But these things do take time, right? Even trials themselves can take 5 years just to go through phase 1 to phase 3 and recruit enough patients and enough time. With Parkinson's disease, of course, you have to look at the progression of the disease over time, and it can take 1 to 2 years for the disease to worsen. So you have to see if the therapy is having a protective effect.
Great, thank you. A question came in about movement and exercise for patients with Parkinson's disease, specifically total resistance exercise and a couple other examples. And I know this may not be your area of expertise, being researchers and scientists, but are there benefits of movement and exercise to those with Parkinson's? And maybe if you can elaborate on that just a little bit.
I'll give that one to Mike.
Okay.
Yeah, sure. So this is actually one thing that's really interesting about Parkinson's disease is that while it's a movement disorder, which makes us, it makes it harder to move and harder for those patients to do their daily activities, there's been a lot of research now to suggest that, moving in some way, going to a dance class, riding your bicycle, these are all productive and great ways to alleviate the symptoms of Parkinson's disease. So certainly a lot of research shows that exercise is beneficial for disease. The specific type of exercise, I think, may be more unique to each patient. So what type of exercise you get certainly could differ from person to person, but exercise is important, and it's definitely been shown to be beneficial for Parkinson's disease.
Great, fantastic, thank you. A specific question about environmental exposures as contributors to Parkinson's. The attendee said, I lived for 3 years inside a geography that ended up as a US Superfund site for cleanup of tetrachloroethene, PCE. Is that a potential factor of Parkinson's, a risk factor or an environmental factor?
Yeah, potentially. I mean, we don't really know about how much you would need to be exposed to and for how long. So it's very difficult for us to say, you know, answer this particular question, like if you live in a certain area and, you know, what your risk would look like. It's important to know how these studies are done. And this is kind of epidemiology, right? So they look at the number of people that have Parkinson's disease in a certain region and versus other regions that don't have certain, you know, toxins. And they try to make associations between the presence of a toxin or some chemical in the environment and the number of people, the incidence of Parkinson's. So you never have the causality there, right? You never say this chemical causes Parkinson's. You just have a higher incidence in that region. That's how all epidemiology is done. It's very unsatisfying, but this is what it is. So I don't have a good answer for that definitively, but sure, you know, probably not a great thing to be exposed to, definitely.
Yeah, but I would, I'd follow up on Darren's point that this is one of the great things about the MIND initiative, the new MIND program, right? Is that if there are exposures, for example, in Western Michigan, we haven't really had the opportunity to track that and track the patients associated with potential exposure. So this really gives us the opportunity. There's a lot of agriculture in Michigan to really assess if any of these, if we see any signatures in the biofluids or tissues that we collect that could be related to those exposures.
Yeah, that's a great point.
Thank you. Dr. Henderson, in your research, has anything shocked you or surprised you as you moved along? Any like aha moments as you go through the research cycle?
Yeah, well, there's a lot of, I mean, we're here every day and discover something new, right? So this is why we come to work. I think it's a lot of things that are exciting. One thing I can say is that that's been really exciting for me is that for a long time, there's been this hypothesis that proteins can spread through the brain, possibly through the neuronal connections. There's been a lot of debate about this because it's really difficult to show. You can't show this in humans because we only look at end stage. Even in our different models, it's been difficult to show. So one of the things that was really exciting for me is that we developed a method to track these protein pathologies over time in animal brains, and then partner with computational neuroscientists and really assess, these different hypotheses. Does pathology spread through the brain? Are there just certain regions that are more vulnerable or resistant? And so this was really a gratifying part of my research when we identified that there was a major contribution of spread through neuronal connections of these protein pathologies, because that gives us a new insight into disease and a new potential avenues for where we can target therapeutics and when during disease certain things might be efficacious. For example, if pathology is only in certain regions early in disease, maybe we can slow progression simply by stopping that transmission or the spread of pathology. So that was really gratifying for me. It's something I was excited about and research that we continue now.
Thanks. Dr. Moore, any surprises as you've gone through your career? I know it's a loaded question.
No, I mean, nothing really surprises me anymore about this disease, but okay. I guess what I want people to understand is that we tend not to be surprised so much because science is a very slow and painful process, right? So it's incremental. So typically you don't come in one day and something massive is discovered. It happens over time, right? But I would say, in terms of the way Parkinson's research has gone, I'm kind of surprised about the sheer number of genes that seem to be linked to disease, and the fact that some of them are implicated in both the familial and the sporadic disease. And I think we all gambled on this in the very beginning, that familial genes, if we could understand them, would be the key to understanding sporadic disease. But now you start seeing genes like LRRK2 and alpha-synuclein, which are actually involved in the risk of sporadic disease and cause familial disease. So this just kind of, sort of solidifies that hypothesis and that gamble that, if we can make drugs for these two proteins, at least they should apply to everybody. That's I think, probably one of the things that surprised me. I kind of wanted it to be the case, but I'm happy that it is. So.
Have time for a couple more questions. One that just came through. Is it safe to assume that I inherited my Parkinson's disease from my uncle, and should my daughter have genetic testing? Very specific question.
Right. I would say it's not safe to assume that you inherited anything from your uncle. I mean, you have to remember these diseases are quite common. So in any family, you're going to know somebody that may have Parkinson's or may have Alzheimer's. So I don't think you can make that jump. But now I think if you can get a family tree together, start seeing that in each generation, a certain number of people had Parkinson's, then that's when we start to suspect inheritance, right? But if it's just uncle to, to you, then I'm not sure we can make that jump. You might want to ask around about other family members, more extended family members, whether they have it too.
Good, fantastic. Do not see any other questions. One last question for each of the doctors. Dr. Moore, what got you started? Why Parkinson's disease? Why neurodegenerative science? What was your driver going through school that you wanted to focus on that?
Right. I think for me, it was a little bit of serendipitous. I kind of stumbled into this field a little bit. I'm glad I did. It's been a very challenging career so far, but I did my PhD on basic neuroscience, so no disease focus whatsoever. And I was very frustrated at the things I was working on and discovering had no real disease implications. And so I just, the next stage of my career, I decided I kind of dabbled in Parkinson's a bit, and I think just fascinated me, and all the genes were coming through at that point. So this has been a great career to have, and a great challenge. And I feel now very passionately about Parkinson's, and I couldn't imagine working on anything else. So.
Great, thank you. And Dr. Henderson, how about you? What was the driver for you to focus on this particular research area?
Yeah, so I've been interested in the world around me, in science for a long time. And so like Darren, I kind of got into school, decided to research neuroscience because the brain is fascinating to me. But then as a grad student, when I looked at the different things that I could research, it really became apparent to me that there's no disease-modifying treatments for any of these diseases, and this is a major place where biomedical research could have an impact. So in grad school, I decided to join a lab doing neurodegenerative disease research, and I haven't looked back since then. We look across multiple neurodegenerative diseases. Like Darren said, these may have common underlying mechanisms. So I'm really fascinated by Alzheimer's disease, dementia with Lewy bodies, Parkinson's disease, and really think that this is a place where biomedical research could have an impact. So that's why, for me, I've also joined this field, and I'm very excited by where it's going.
Just to follow up on that quickly, Mike and I are in the business of trying to put ourselves out of business, right, Mike?
Yep.
Yes.
There's plenty of diseases to work on, so we hope we don't have to work on this one for too long. Definitely.
Well said, thank you. And I do not see any other questions, and we are almost at the end of our time. So thank you everyone for joining us today. Thank you, Dr. Moore and Dr. Henderson, for your time and your insights, and just fascinating background in what's going on at the Institute. Many of you on this call today generously support our world-class Parkinson's research, and just on behalf of our 500 employees at Van Andel Institute, representing over 40 countries, thank you so very, very much for your support. One thing I just wanted to mention, when you donate to Van Andel Institute, 100% of your donation goes right into research in our Parkinson's labs, which is unprecedented across the research spectrum. 100% goes right to the labs. So we invite you to reach out and visit us for a tour, or financially partner with us as we continue the fight against Parkinson's. After we conclude today, you'll receive an email including a link to support our Parkinson's work, as well as an opportunity to complete a survey providing feedback on today's event. So thank you for your consideration. Our next virtual public lecture will be held on December 9, just a couple of months away. We'll focus on today's educational landscape and the innovative efforts taking place at Van Andel Institute's Institute for Education, supporting our teachers and our students. So once again, thank you, Dr. Moore. Thank you, Dr. Henderson. Thank you all for attending today, and have a great rest of the day. Thank you.